We at the Department of Nanobiology at the Cancer Research Institute Biomedical Research Center in Bratislava, we focus, among others, also on nanotoxicology, as it is essential in estimation of side effects of potential nanomedicines. So nanomedicines offers a unique opportunity to combine diagnostics and therapy, so-called Theranostics, into one therapeutic agent. And nowadays, the therapeutic agents are countless number of nanoparticles of different shapes and sizes, and core materials and surface modifications and so on. However, for all of them, it would be very useful if after they fulfill their therapeutic mission in the body, they could be effectively removed from the body. The most effective way would be their removal by blood filtrations in a kidney. However, renal filter has certain limitations. What about size and shape of material filtered? Therefore, these would need to be considered very specifically for each nanomaterial. Nevertheless, there always is a possibility that nanomaterials reach renal cells regardless of being filtered or not, simply thanks to blood circulation. Therefore, one of our research aims was to investigate impact of magnetite nanoparticles. We already have heard that they are very promising in Theranostics. So magnetite nanoparticles, spherical, with 10 nanometer core, coated with polyethylene glycol, PEG, or bovine serum albumin, BSA, on highly specialized, very unique renal cells that are involved in blood filtration in the kidney. So they also reside at the site of blood filtration in the kidney glomerulus, and these cells are called podocytes. Here in green, podocytes are essential part of filtration barrier, and if their function is disturbed, it may lead to nephrotic syndrome, a very serious condition. For our experiments, we did not use commercial cell lines, but we rather isolated primary podocytes directly from mouse kidneys using magnetic beads. So these are not nanoparticles.
I'm very busy. I got confused. Today's day 1. Sorry. Tomorrow day 2. Okay. Thank you. Sorry.
So these beads are not nanoparticles, but they are just magnetic microbeads used for isolation. Certainly, we characterized outgrown cells in the culture, and we checked to be podocytes, and we checked for specific podocytes marker proteins like Wilm tumor, one like nephrin, synaptopodin, podocin by staining and by RT-PCR as well. And once the cells were characterized, we exposed them to nanoparticles. And we observed a very different inflammatory profile these nanoparticles induced in podocytes. Just to note, the nanoparticles differed only in the outer coating. So while BSA-coated nanoparticles induced rather early inflammatory response detected 5 hours after exposure or none, pegylated nanoparticles induced rather late inflammatory response detected 24 hours after exposure. Here we checked for expression of pro-inflammatory cytokines like TNF-alpha, IL-6, IL-1 beta, and so on, or pro-inflammatory chemokines. This was also mirrored at the protein level. Secretion of TNF-alpha and IL-6 was strongly increased after exposure to pegylated nanoparticles after 24 hours, but was very low after exposure to BSA-coated nanoparticles. The same was true for other inflammatory factors like INOS. Again, for pegylated nanoparticles, late inflammatory response and increased level of protein, and for BSA-coated nanoparticles, early inflammatory response and no proteins. From this data, one would assume that BSA-coated magnetite nanoparticles might be less toxic, less harmful for podocytes? Well, we thought that too, unless we checked for another important factor, that is change of cell shape induced by nanoparticles. Unfortunately, both types of magnetite nanoparticles, PEG and BSA, activated actin cytoskeleton remodeling in podocytes. Here you can see control cells and actin network in red, and focal adhesion molecule vinculin in green, and cells 24 hours after exposure to PEG and BSA-coated nanoparticles, and you see a dramatic change in actin network as well as in adhesion properties. Why this is so important for podocytes? Podocytes in the glomerulus cover and surround blood capillaries very tightly with their food processes, and this ensures for proper blood filtration. If any substance change this highly specific actin structure, the podocyte food processes change, and this is so-called food process effacement, and this is already sign of podocyte injury. So actually any change in actin cytoskeleton is considered to be podocyte injury, sign of podocyte injury, and it has consequences. Injured podocytes cannot build a proper barrier for filtration, and so proteins from the blood can leak into urine, so we end up with proteinuria. If this condition lasts long enough, podocytes can detach completely from the blood capillary and can be also washed away with urine. So their place on the capillary stays empty, because once podocytes are lost, they are lost forever and cannot be ever replaced again. Therefore, it is very important for any substance, not just nanotherapeutics, to check for these effects. We also quantified number of nanoparticles podocytes internalized, and to our surprise, we observed that podocytes are able to internalize much higher amounts of nanoparticles than other renal cells, for example, here mesangial cells. We also were curious where these nanoparticles are stored within the cell. So with the aid of electron microscopy, we identified and detected nanoparticles in lysosomes, in vacuoles, what would be kind of expected, and this accumulation was associated with damage of cellular structures like Golgi apparatus, like mitochondria, like endoplasmic, rough endoplasmic reticulum, and so on. But for pegylated, and only for pegylated nanoparticles we saw this, there were additionally huge vesicles filled exclusively with the nanomaterial, and cells carrying these vesicles displayed even higher signs of damage than the other cells. Well, these were just few findings I could present, what we estimated for magnetite nanoparticles and renal cells. Who would like to discover more? It would be found in our publication by Schultz et al., published by Royal Society of Chemistry in RSC Advances last year. So please feel free to check. And with this, I would like to thank you all for your attention.
Thank you so much for your presentation. Questions, please? If you have questions, don't hesitate to use raise hand tool. Okay, I have one. Could you please comment on the possible mechanism of the podocyte damage caused by the magnetite nanoparticles? Is it a production of the reactive oxygen species?
Partially it might be, but surely it is not a major mechanism. What we learned from electron microscopy was that the cellular damage per se was due to presence of these magnetite nanoparticles in the cell, because the damage for mitochondria and especially Golgi apparatus is deadly for cells because they are important structures involved in cell well-being and living and working. So this is from the mechanistic view, but for the others, we really detected expression of inflammatory factors. So I'd rather say that the major mechanisms would be initiation of pro-inflammatory response, but not just by ROS induction, but just by activation of signaling pathways of inflammatory, activation of inflammatory signaling pathways.
Have you any data on the apoptosis induction?
We also checked for apoptosis, but rather like apoptosis proliferation assays at the beginning, because we needed to set up the concentration of nanoparticles used for experiments, and we did not want to use already toxic concentrations. So there we saw that the response of podocytes was somehow similar for high concentrations, but these concentrations, what we used for these experiments, were not toxic and did not activate apoptotic pathways, so were not somehow into apoptotic direction, but really more to the inflammatory direction.
Okay, thank you very much.